Bismillahirrahmanirrahim, and welcome back to this next video. And in this particular video I am going to tell you about the difference between the nucleoside and the deoxynucleoside. So the first thing you need to understand what is a nucleoside. When you talk about the nucleoside, it is actually the sugar attached to a base. So this is simply called is the nucleoside. And when a phosphate group that is attached to a nucleoside, it is converted into a nucleotide. I have a detailed video on the structure of the DNA, and I'll share the link in the description. But for this particular video, just keep in mind that the sugar and the base, when they are connected to each other, it is called is the nucleoside. And for nucleoside, it can be a simple nucleoside or it can be deoxynucleoside. So what is the difference between the nucleoside and the deoxynucleoside? Then the difference lies in the sugar. So when the sugar that is in the ribose form, by that I mean that there is a hydroxyl group at position number 2 as well as position number 3. We usually call this is the ribose sugar. But when the hydroxyl group of the position number 2 that is removed and that is replaced by a hydrogen group, we call it as the deoxyribose. So when the sugar that is a ribose, we call it is the nucleoside, and when the sugar that is deoxyribose, we call it is the deoxynucleoside. One of the important mistake that the students usually make is that they are not able to differentiate between the adenine and the adenosine. So you need to understand this. When we use the term adenine, we are simply referring to a purine base. Nothing is attached to that. But when we use the term adenosine, that means that sugar that has been attached to the adenine. So if the sugar that is ribose sugar, we call it is the adenosine. But if the sugar that is a deoxyribose, we call it is the deoxyadenosine because of the sugar, because that is deoxy in nature. Similarly, when you talk about the guanine, this guanine term is simply the base guanine, which is from the purine family. But when a sugar that is attached to the guanine, and if that sugar is a ribose sugar, we call it is the guanosine. But if that sugar is deoxyribose, we call it is the deoxyguanosine. And the adenine and the guanine, they both belong to the family of the purines, which are actually two rings that are fused to each other. When you talk about the cytosine is actually a base. So when the ribose sugar that is attached to the cytosine, we call it is the cytidine. But if a deoxyribose that is attached to the cytosine, we call it is the deoxycytidine. The uracil, again, this is a base, and when ribose sugar that is attached to the uracil, that is called is the uridine. This uracil is usually not found in the DNA, so we do not actually have any terminology for the deoxy form. When you talk about the thymine, this thymine again is usually not found in the RNA, but it is present in the DNA. That means that thymine that will be the base, and when the deoxyribose that is attached to it, we call it is the thymidine or the deoxythymidine. Now because this thymine is usually not found in the RNA, so this deoxy designation for its nucleoside is frequently assumed, and the deoxynucleoside is simply called is the thymidine. So whenever you see the term thymidine, it simply means that you are talking about the deoxy form. So this deoxythymidine or the thymidine, they are actually referring to one in the same thing. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.